today. So I would like you to have a little bit of an overview of because we've been talking a lot about obviously SARS-CoV-2 and coronaviruses, and to a certain extent that's rightly so. We are through a pandemic, but my a little bit of provocative question today is to try to think a little bit wider and to look at what's going on under the surface of this boiling sort of life that is the sort of the viruses and how in reality we've got so many that are there, uh, well known, some others less known, and what kind of interaction we've got with them. And so to frame a little bit the, the picture in a wider context. So if we start from the beginning, which is basically what is a virus? That's for some might be a simple question, and you could answer that is a very, very small organism, like really small, probably the smaller organism that we know, and we we know that they are infectious. They are basically transmitted via infections, and but what is true is that they are so small that we carry billions of these particles on our body, and actually we could think that a single person would carry billions of viruses of SARS-CoV-2. So it's a remarkable number of particle viruses. But yet, if you put them together, all these viruses present in a person who is infectious for SARS-CoV-2, the mass would not exceed 0.1 milligram. So such a small amount, not even a gram, not even a fraction of a gram, is 10,000 smaller than a gram. So, in fact, if we could think about all the SARS-CoV-2 viruses in the world that we know they are and infecting the people and the million of people, probably we wouldn't get more than kilograms. So these particles are actually incredibly small. We know that, for example, if this is a bacteria, and you know bacteria quite well, and actually probably some of you have seen them in at the microscope in the lab using a 100 times magnification, and you can see some of these bacteria on your microscope. There are some viruses recently discovered that they seem a little bit bigger. Actually, at the beginning, they were thought to be bacteria. One that is particularly interesting because of the name is the mimi virus. Mimi, like mimicking, because at the beginning they thought they were bacteria and they sort of mistook them for bacteria. Most of these big viruses infect amoeba, like protozoan, like cellular eukaryote. But if we go smaller and smaller, we get to the kind of known viruses that generally infect humans. Here we got the HIV, that is the virus causing AIDS, the Zika virus. Some of you, well, we will talk about that in a moment, the virus that was causing a lot of trouble in Brazil and during the Olympic Games in 2016. And then the parvoviruses. Parvo means small, and they are really, really fraction of a bacteria. So this is a kind of a real representation of the size of these organisms. What do we know about diversity as well? We start learning and learning more and more as the science progresses. And in the last 20 years, this knowledge expanded so much. And I invite you, for example, to have a look in this link. This is an expedition, is a project which is called the Global Ocean Sampling Expedition that is headed by Craig Venter, is the guy who, together with others, basically sequenced at the beginning the human genome in 2001. And now what he does, he goes around the world sampling, and he is sort of doing the route of Darwin, and he samples the variety of life in water. And what he has found is that there is a myriad, an incredible amount of viruses that are floating around our waters we didn't know about, but they are essential for feeding plankton and natural life, and also to produce phosphorus and carbon that are essential for life in the oceans. So we're learning more and more about the incredible amount of viruses that we normally live in. And when you go in the sea and you swim and you take a sip of water, you probably are ingesting millions of viruses in that simple scoop of water. We know that viruses are not only terrible organisms, actually, rather the opposite. We live together with viruses. Most of these viruses are completely irrelevant to us. Like when you eat your veggie, for example, there are so many viruses that are normally covering some are infecting the plants, some are just covering on the surface of the plants, but they're not even able to infect us. They just go through our system and then with no consequences at all. Instead, we got several viruses, many viruses that are normally found in our body. And this depends on the kind of district we are looking at in our lungs, on our skin, in our guts. Some of these are symbioses, like they are normally there, and we would call them symbiosis in a way that they live with us with no particular effect. Some, instead of other interaction, are more antagonistic. So they can cause conflict with the host, and they can infect us with clinical relevance. We know, for example, SARS-CoV-2 is an example, but we know measles, we know so many other viruses that some of them we will look together during the course of this presentation. And there are different forms of interaction. We talk about symbiosis, we talk about antagonistic. There are also some mutualistic interactions. For example, Salmonella and some phages, which are these sort of viruses here that they look a little bit like a robot with legs, and here is a head, and then they drill through the bacteria with this part here, and they inject their genome inside, and they can do their viral cycle. Very clever. But some of them, they are actually inducing an advantage in the host. And then we've got symbiogenesis or endogenized viruses. So you might know or not that almost 10% of your DNA is actually derived from viruses that are ancient viruses, that they colonized our body, they integrated into the DNA, and now they've got some functions. For example, some endogenized viruses that now they are essential for the formation of the placenta in the human being. So it's a remarkable amount of viruses that interact with us in different forms, in different relationships, some mutualistic, some symbiogenetic, and with proper function in our metabolism, some more in conflict, yes. But let's see how this is panning out and how is it working. So I ask you a question, and please feel free to interact with me. And I would like actually to ask this question to you, and I hope you feel free to answer this, which is, what do you think about a virus? Is it alive? Do you think a virus is an alive organism or not? And I can tell you already, it's not an answer that many will have the same view and the same answer exactly. So, what do you think about this question? If you can unmute yourself, I'm very happy for you to do this. It's just your opinion on this. Do you think a virus is a live particle? Don't be shy. If like, but I can help you with that. So if you think there is a common answer that everybody will give you the same answer, there isn't. Okay, a virus is a organism that is in between. It's difficult to categorize it as a live form of in the biological spectrum, and I will tell you in a moment why. We need to analyze the life cycle in order to understand whether virus is alive or maybe it's an in between. So viral genomes and morphologies, these are the main elements that distinguish a virus from another: their genome, their genetic components, and the shape of them. Here I give you three examples. Examples is a SARS-CoV-2 that now we know, an EBV Epstein-Barr virus is a DNA virus. This is an RNA virus, different genome form, and an Ebola virus, quite dangerous, but luckily not very infectious in spreading from human to human. So very different forms, as you can see, okay? They all have got a surface that has got this sort of bullets, this sort of needles coming out. We will learn that these are ligands searching for something that is called receptor on the surface of our cells. And this is important for the mechanism of key and a keyhole in order, or lock, in order to open a door, basically. And the door is the door of access to the cell. So the mechanism by which this virus will gain access to our cells is by finding these ligands that they can bind efficiently the receptor of the cell. And they all have got them. You can see very clearly here. They are called glycoprotein, but it doesn't matter. All of these viruses, then they've got a protection, a sort of tegument, which is called capsid, made of protein, very strong. And then they've got the genome. This is the genome inside, okay? The genome can be a whole one, can be fragmented, can be DNA, can be RNA. And what is important is mainly these two elements: their nature of the genome and the nature of their capsid and envelope. So the form, the shape, and the cover and the genome they've got. These are the two main descriptors. Then they will diversify, they will distinguish a virus from another. And in terms of nature of these viruses, we call it, you might hear about species, okay? In animal kingdom, there are species of different carnivores and species of most animals. But with a virus, this is not easy. We call it quasi-species or almost species. So what is it? It is that most of these viruses, yes, you can distinguish them one from another, but what happens is that their nature, basically their genome is constantly mutating, is changing. And it changes so much that often you start seeing a variety of different viruses that they all are basically in the same genre, in the same family, but they sort of specify, but they are quite different. So they call it quasi-species. And this is the reason why viruses, they constantly change to try to get access to this different receptor more and more efficient, and to sort of evade the surveillance of the immune system of the host. So they're quite variable organisms. This is a complex slide, don't worry about that, but this is just to tell you that the genome, we use their genome to basically distinguish them. And these are the main 7 categories that we are using: DNA, plus RNA, and plus and minus, and all the other. I don't go into detail of this, but we know there are different 7 main categories that are distinguished by the genome, and this is called the Baltimore system of classification. What is important about this? Well, it's important because at least it gives you an idea in order to group different viruses and to sort of distinguish them one from another. Inside these categories, you will find different quasi-species, and this will be the greatest variety of virus that you will see. And then it's a sort of quite interesting experiment you are verifying now under your eyes with SARS-CoV-2. You know it's a coronavirus, yes. You know it's a SARS-CoV-2. But then you start seeing the alpha, the beta, the delta, the Omicron variant. And this is the classic example. You are seeing under your eyes evolution happening and what we mean for quasi-species. They are different enough because you can see how the delta is more dangerous than the alpha and is spread quite quickly. But then there you go, the Omicron, which is even more infectious, but is less clinically relevant. Still is the same SARS-CoV-2. So in order to understand a virus, and the question that I've asked you, is it alive or not? Well, I just told you that a virus per se is a particle that has got a genome, a tegument, a sort of capsid of protein that protects it, and then this ligand, this glycoprotein that they are looking for a receptor to enter in a cell. This is called tropism. This cell is called susceptible. It means it can be infected, and if it allows for the full viral cycle, which is entering the cell and multiplication outside the cell, is also called permissive. Okay, but what is important here? The important factor is that a virion, which is a virus outside the cell, is just, as I told you, a genome with protein outside, which potentially can infect a cell, but can't do much. It cannot do anything else than floating around and trying to pass to a new cell. In order to do a full life cycle and to multiply, it needs to get into the cell where the cell will give all the full set of enzymes and protein necessary to induce a full life cycle and therefore to multiply. Now, if we think that life is propagating and multiplying, that's okay. This is a live organism. But in order to be alive, it needs a cell to infect. And therefore, we call it an obligate parasite. So you have a stage outside the cell, which is definitely not very much looking like an alive organism. But when it gains access to the cell and it forms a full set of other particles that you could see many other particle viruses forming here and then emerging from the cell. This is the cytoplasm of the cell. So this is the same cell. It shows the virus that is entering, then multiplying via different mechanism. Oh, sorry. And then aggressing exit in different ways. That generally called budding. And here you have millions of viral particles from one. Now, this is a full life cycle, and this would be called, let's say, an alive organism, okay, a living organism. Now, it's important to know that all viruses have got the same stages in viral infection, which is entry. They go an entry point, then they fuse, So the entry is basically the binding with the glycoprotein to the receptor. Then they fuse with the membrane, they enter and they inject their viral genome. At this point, they can replicate. So they've got more viral genomes, and then they start producing an mRNA that will produce proteins. So these proteins, they are essential for producing new capsid, so new structure around new glycoprotein. So eventually you will have millions of these new viruses that will emerge and will go and infect a new host. So this is the viral cycle, and all viruses have got this. This is an example, for example, of the HIV. And in HIV, you see the same thing happening. It's got the same viral cycle. And remember these phases: viral entry, replication, assembly, aggression or budding, emergence from the cells, because I will take this back at the end when we look at drug intervention. This is an example of an infection of HIV, for example, which has got the same viral cycle. Viruses have got also phases. Generally, it means that when we talk about phases, it means phases of infection. We are not talking about the viral cycle in the cell, but in their interaction with the host. You know, because if you had the virus, what happened at the beginning? Well, at the beginning, you don't even realize. Then you feel a little bit rough. And then you start having symptoms, and then hopefully there is remission, convalescence, and you feel better, and you develop maybe an immunity. With HIV, it's a little bit more complicated because, yes, there is an acute infection where the virus, you can see here, the viral load means the amount of virus in the blood is the red thing. It gets really high, and people start having a little bit of symptoms, not very strong, but maybe like a flu. And then it goes into an asymptomatic phase where the virus is actually controlled by the organism. And then slowly the virus wins its battle with the immune system and it starts re-emerging very strongly. And this is the symptomatic phase, or the immunodeficiency syndrome, where it's called basically AIDS, and it can lead to death unless it's treated very early with specific drugs. Most viruses have got a very similar sequence of phases. If we look at hemorrhagic fever viruses like Ebola or dengue or Lassa viruses, they've got a similar stage where you got a prodromal phase where you got some symptoms, and then flu-like symptoms. And then if we get worse, you go into hemorrhagic phase. And then luckily for some, you can get into convalescent phase where you get in remission and the symptoms disappear, you form an immunity and you get better. In some cases, instead, it leads to death. So having said what is a virus, how it divides, what is a life cycle, and also, I would say very importantly, what are the phases and the clinical signs in this sort of conflict. This is a more antagonistic interaction with viruses that we've looked at now. The second part is more asking, okay, we know about SARS-CoV-2, we do know, maybe we know about flu as well, but is that all? No, it's not. And we should define why this is not the case and what we know already about so many other viruses that are around, and they are ready to do the same thing, to jump in and out of a reservoir. It could be an animal into the human, back to the animal, and from human to human. Now we start talking about a more complicated but very interesting and fascinating mechanism, that is the zoonosis. So I call this so very far, so very close, because we know often that we listen to the radio, we look at TV, we watch movie, we read newspapers, and like it's 2 days ago, 3 days ago, maybe you've read this, maybe not, but there was a case of Lassa fever in Bedfordshire, and actually I don't know how it went in terms of spreading, because I know that the whole family then eventually was infected, and there was definitely at least one victim. The Lassa fever is an hemorrhagic fever. It's typical of Africa. It's not very it's very clinically relevant. It's very dangerous when you get it, but luckily enough, it doesn't spread very much in the human. They are a kind of dead end. We will see infection. They generally stay in other organisms. They are called reservoir, like bats. They can be mice, depending on the virus. In the case of Lassa fever, sometimes it can get out of the reservoir, getting into the human. This family just flew back from Africa, where they were there for a trip. They came back to London, and they realized after the incubation, if you remember the acute phase or the prodromal phase, which is the initial phase, they started having symptoms, and they realized it was a Lassa fever, absolutely not common in our country. There are, well, we know about SARS-CoV-2. You might have heard about Zika virus during the Olympic Games that put under threat in Brazil the Olympic Games, but also the life of several people in that area. So I could go on and showing you all these viruses. Ebola virus, the Zaire, avian flu, also called H5N1, dengue virus, Zika virus, Endra, MERS, SARS, SARS-CoV-2, Lassa, Junin, West Nile, rabies, antivirus, HIV, swine flu, Marburg virus, and on and on and on. What have got all these viruses, including measles, including smallpox? What have they got all in common? They all came or come from a different reservoir than one that we normally see in the human. So yes, they are all viruses that can infect. They can infect or infect endemically humans, but they all come from a different history. Actually, the virus that we know that they were in human population we are talking about our ancestors. We are going back in the prehistoric time. There are very few of them. Most of the viruses that infect the human, they come from different reservoirs that are actually animals, non-human animals. And then they integrated in a way in our lives, often causing havoc, okay? So we call them emerging viruses. What's an emerging virus? An emerging virus is a new virus, which not previously described. We didn't know about this virus, and now it comes and infects us, unfortunately, like SARS-CoV-2, then the present pandemic. This virus was in bats, and we will see how these things more or less they happen. And then we didn't even know about that. We knew about coronaviruses in bats. We knew about something similar to SARS-CoV-2, but it wasn't dangerous. It wasn't recognized until now, where everybody has to recognize it. Or is a virus that is known, but all at once it shows new clinical features. This is the example of the Zika virus in 2016 in Brazil, where I think the virus was known since the 50s, the 60s, decades, but from Africa, but it wasn't really a concern until it was in these outbreaks in Brazil. It was linked to congenital defects of microcephaly, like basically children born with defects for life, and therefore it was a big concern. And therefore we have to introduce the concept of zoonosis. A zoonosis is it basically describes the route of how it gets to a human, and means like a virus that jump in a way from a non-human to a human host. And then it depends the fate of this virus. If the virus jump, but then doesn't have much of an impact in a way that, well, let's reformulate my sentence because it's not correct and also respectful for the people who get infection. It can be incredibly impactful for the people who get infected. In fact, it can die, or several people, but the virus doesn't have the power to transmit efficiently in the population. So this is called a dead end zoonotic outbreak. Some of these viruses are really terrible, like Ebola virus, Lassa virus, Enterovirus, MERS. I mean, these are hemorrhagic fever viruses, most of them, and they cause incredible pain and clinical signs in patients, which are multi-organ failure, is really bad. And their death rate is sometimes as high as 60%, which means if 10 people get it, 6 will die, and 4 will probably have terrible consequences before they get better. So, but luckily enough, they can't spread very quickly and very efficiently from people, person to person, unless there is a clear medical procedure, like a cut in the skin, like some general during autopsy or during some researching in the lab on, let's say, via handling infectious tissue or blood, but it won't spread just by breathing, okay? Whereas other viruses can establish very quickly, and the zoonotic jump will become in an outbreak, in an epidemic, and often, like it happens, not often or likely, but in a pandemic like with SARS-CoV-2. And please do not forget HIV. HIV, it was possibly a simian virus, so a monkey virus at the beginning, then slowly mutating, getting into human, and now having infecting the whole, when I say the whole world, I mean the whole area of the world, and created more than 37 million deaths. And I think at the moment there are 35, 36 million people positive at the moment on this planet. It keeps causing like a death of around 700, 600,000 per year. It was a very successful virus, unfortunately. Okay. So it's not just SARS-CoV-2. Okay. In terms of zoonosis, this is a very beautiful um, subject that maybe some of you in the future will go study and help with knowing more, understanding it more, and also producing plans and sort of strategies that are better to contain also this sort of events. But first, we need to understand them. A zoonosis, as we said, is a jump from an animal of a virus or a pathogen in general, but today we talk about viruses, from an animal to the human. And this can be a wild animal. Here you got a monkey, a gorilla, mice, and bats. Ooh, those are really, really like gold reservoir, and we will see in a moment why. You can have intermediate hosts like ticks, fleas, arachnids of different kinds. But there are, like, for example, the several tick-related, like encephalitis, that are viruses that they are transported by tick, but they can also bite into mice and then back into ticks, and then they can spill into human. Or they can spill from cattle. So we know about the swine flu, we know about the avian flu into the human. We know about MERS, some of the Middle Eastern coronaviruses, and they can get into the human, or they can get the trajectory from the wild to the pets and cattle to the human. So this graph is describing basically the mechanism of zoonosis. So there are different jumps possible. You can see here the wild animals sort of cycling these viruses in a way of sort of outbreaks in the wild animal population. And then sometimes they can spill into the human directly. One example is rabies that we will do in a moment. We will describe this case study. Or they can spill from the wild animal into the pets and cattle, and then into the human. Rabies is a very good example too. If we remember how SARS-CoV-2 emerged, the most likely is that from a bat, it got into the human population. How? That's not easy to describe at the moment. This is another interesting sort of slide that describe how all these movements happen. And you can see here, you've got like reservoir hosts, and you can have the wild or some cattle here. You got the mice, which are wild, let's say, but they are more related to us in a way. They are kind of they get around quite well. So this depends in first instance the reservoir distribution. So how the reservoir is distributed. So basically, if we think about mice, they can multiply very quickly and get very dense and in close relationship with our houses and with our cattle. So all these are factors that initially are an incentive to initial spread and possible zoonotic jumps. Then we go from different barriers, different levels of sort of roots and barriers to get finally to the man, to the human in general. So you can have, for example, practice like slaughtering. And you know that in slaughterhouses, you have to unfortunately kill the animal, then open it, and then all the interiors, all the blood is in clear contact with the human being, and therefore the butcher, they have to prepare it correctly. And at that point, there is a level of danger if there is a possible infection there, which will derive by obviously all these initial cases of how much this animal mingled and distribute the virus one to another. And then obviously how much of these animals are harvested constantly or not for feeding, for example, and how they are transported around and the sort of hygiene that is got used or not. And these are sort of funnels that eventually will lead to a dose exposure higher or less to the human population, and therefore will increase the dangers of human infection or zoonosis. And you can do this for all sorts of different routes. There are different routes, different level of separation, different barriers, and you can analyze all of this epidemiologically. And this is a work that you would do if you are doing any surveillance, zoonotic surveillance as a scientist. So in summary, we spoke about the zoonosis, what it is, how it goes around. So it's a jump from an animal to a human. And we spoke about the factors that can improve this from happening. And also the way of transmission. And I would say that one thing we haven't mentioned is these are all factors that have to do with the environmental pressure, with the environmental constrictions, but there are also factors that are more intrinsic to the virus itself. So having in place all these parameters, then there is the variability of the virus. And as you know, for example, with SARS-CoV-2, you can make your best to try to remove or to implement barriers in these environmental conditions. But then if a virus mutates and has got a very sustained transmission, a very high infection rate, then obviously you can put mitigating factor in place, but it will get around no matter what, especially if it's a respiratory virus. So now I would like to, because we said most of the viruses are actually zoonotic viruses. We spoke about smallpox, that actually smallpox is an interesting subject, whereas it was possibly moved from camels, which are actually originated from North America. They were moved to Africa. And then they got infected with the smallpox from gerbils in the African region, which coexisted in the area where camels were. And basically what they did then, because of human using camels for transport and everything, they start jumping from camels to human, and then smallpox did its first zoonotic appearance. And then when it was transported, it was moved by just movement of people all around Europe. And then when the European colonized America, and so they brought it back to America, it was an incredible disaster, because obviously the population was naive, never seen this virus, and there was death of millions of, for example, the Aztec population. Similar thing happened with measles, that we know today infecting mainly children, but it was a spillover following domestication of animals during when the human beings started farming, and then having sort of animals around for food and for work. And they spilled from these cattle to the human. And then following urbanization, which pushed the human together, it spread all over. These are pictures of what can happen when a zoonotic virus get around. Like this is an example of the swine flu, or the foot and mouth, and where, I mean, millions of carcasses were piled and burned because of the, not just the threat that they've got to the human being, which is in certain cases not even that high, but is basically the waste of the meat, that it becomes unusable. And this is very sad, incredibly sad image to see in that period. Now let's talk to finish, because I want to talk in the last 10 minutes about case studies, and particularly I want to give you 3 examples of how these viruses get around. And in order to understand them, we talk about 2 main reservoirs. One is the rodents, little mice, and the other one is bats. So these are flying mammalians. We know about, for example, horseshoe bats. You might have heard about that because of the SARS-CoV-2. Now, if we look at the distribution of some of these bats that are known to carry hundreds of viruses, and actually we found thousands of new viruses, some of which potentially dangerous. And you look at the distribution around the world, you can realize that, well, they're quite everywhere, in a way that they could create a spillover or a zoonotic events a bit everywhere. So luckily enough, this doesn't happen. Why? Well, one of the reasons why is that despite the bats being there, despite the mice even more everywhere, now we are looking at these dangerous, potentially dangerous bats, which are beautiful, and they are actually sentinels that show that the environment is in a good shape. So it's good to have bats, but they are potentially involved with transmission of zoonosis. So why is this happening not everywhere? Because part of the zoonosis, as we said, is not just the virus that is infecting the animal, but is also the selective pressure that is given via environmental changes. These are climate change, these are deforestation, these are practice like food market, which don't respect certain basic hygiene rules, and so on and so on. So luckily enough, if most of these other elements are in place, these these events are not that recurrent, and therefore we don't see them very often. But they are there, ready to happen. And so that's why we should really take care of the environment. We should really take care of the forest and make sure we don't push the animal to the limits of their minimal living environment. This, if you take bats and mice and you sequence them, and you sequence, excuse me, the DNA containing their secretions, like you do a swab now to check for your SARS-CoV-2, if you're positive or negative, you find, look at the numbers that they found, incredible, remarkable numbers. It's like thousands of kind of viruses, okay? Thousands, and then 4,000 coronavirus. I mean, SARS-CoV-2 is one of them, but there are actually at least almost 4,000 already sequenced in bats and mice. And they could possibly be of a certain danger, some of them, okay? In order to do this, it's a very recent science. It's not an easy science because you need to put these trapped nets, and then the bats will gently be collected in these nets. You don't want to hurt them. Then you have to swab them like you do, and this is dangerous, okay? You don't want to be bitten because you don't want to be infected. It's dangerous for you. And then you have to sequence their DNA on machines. And this is how you can see how very well protected they are. But how this science of surveillance is surveillance, virus surveillance, has developed in the last few years very successfully, and it's very useful to our knowledge, and it will be more and more in the future. So I'm going to give you 3 case studies before we finish. The first is rabies. So rabies is endemic in wild animals. Here is a skunk, there are bats, there are foxes. These animals do carry rabies. The rabies virus is a sort of bullet shape. You can see it here, quite interesting. Again, it's got this cover of proteins and the genomic RNA inside. These are the glycoprotein that they are sort of spiking outside to look for a receptor. Well, okay, they can multiply in the wild animal, and they can sometimes jump to the pets, like our cats, our dogs, because they come and pee in their area of playing, or we take them out, and sometimes they put in their mouth things they shouldn't, and they just get infected with rabies, and then they can spill over into the human. Or it can be a person working in the wild wilderness, or a vet, and being bitten directly by one of these animals, who come into contact with it. There are practice where a human being, they do come into contact with the pellets and from this animal more often, and it can happen that they get infected. Unfortunately, if this happens, this virus has developed this very peculiar clinical characterization where basically, in order to improve the way to spread, it starts shedding mostly in the salivary glands in your mouth, and then it induces aggressive biting behavior in the host. So that's why it's called rabies, like anger from Latin. And then you can get even more likely to be bitten and therefore to carry finally the virus. The way to stop this from happening is it's very clever what we've done. We're creating vaccines, but not really for the people, which some of us are actually vaccinated, especially vets and people like that, more at risk. But actually, we vaccinate our pets. So by creating now a barrier between the wild and the population, we have stamped out most of the rabies. So rabies is very rare in our populations, Western country where most of our pets are vaccinated and we don't constantly live in close relationship with the wilderness. This is another case study, very interesting, of a virus that it has spread in Central America, and it's really interesting because I think I jumped one. Is the Machupo virus, or is another hemorrhagic virus like Ebola to a certain extent, that was identified in 1963, and at the beginning wasn't very clear what was happening there, and it had a mortality rate of 30%. This was in Bolivia, okay, in Central America. It wasn't very clear what was going on there, and it took quite a lot to understand that. So it similar outbreaks like this, you might hear about the Hunin virus, the Lassa virus we just spoke about, Machupo, Ebola, Marburg, dengue. They've got very high mortality but low ability to spread. But when the outbreak is powerful and when the frequency of interaction between, if you go back, sorry guys, I just move back to one slide where we were talking about the initial reservoir, where this is basically there is a lot of contact. The climate, the environment favors explosion of the population of this reservoir and contacts. And then also for some reason, the conditions are also favorable for interaction between this reservoir and the human. At that point, there is potential for the zoonosis. And this is what happened in Bolivia. And at the beginning, it wasn't clear what it was, but this Calomys was this sort of species of rodent was the main culprit. And now I put in sparse order all the elements that these scientists puzzled together, and some of them got infected, some of them actually died researching this. Incredible and remarkable how scientists are always in first line. And what happened is basically, well, these people had to grow the crop, and in order to grow crop, and at the same time contain a sort of raise in um, mosquitoes in the area and to stop malaria. So to basically decrease the population of insects, to favor crops, but also to reduce malaria, they used a poison, a toxin, which is called DTT, which is very toxic in a high dose for arthropods and insects. But actually, they use so much that they start filtering through the water. And what happens is that they can be dangerous for human beings as well. But cat population, they start drinking the water. And because they are less resilient than other animals, they start dying. Because of the death of so many cats, the mice population exploded. And these mice, they carry this unknown Machupo virus, which is an hemorrhagic virus previously unknown. But because they grow so much, and now the human beings are harvesting, the people are harvesting so much crop, and the conditions are not perfectly hygienic. They put in their house, there is no separation between the crop and their houses. That basically the mice enter, they eat, they feed, they urinate on it, they poo on it. And when the people basically harvest and they come into contact with the dust and the mites and the sort of carrying, lifting all this in the air, all these residues of urines, including the virus, they breathe it and they get infected. And this is how the outbreak worked. Basically, the outbreak and then the epidemic was due to a change in epidemiological condition and environmental condition by uh, basically due to influence of the human being on the environment by poisoning not only the insects but also the land, and therefore poisoning their pets, and therefore favoring the explosion of the pests, and therefore then the contagion of their food, and therefore finally the contagion of themselves. Incredible, not remarkable, incredible, these things when you study after. And SARS-CoV-2 pandemic is sort of a mystery so far. What we know is that if you look at the previous slide, well, it's a sort of it's a spillover at the beginning. It is a zoonotic event, and most likely is a horseshoe or but it's not yet clear. You heard about pangolin as intermediate host. It's really not clear how initially it spread all over. At the moment, um, it's likely that this happened thanks to or thanks, not thanks to, the sort of very poor hygienic markets like a Wuhan market, where you can see all sort of meat that is exposed with no hygienic barriers and rules, and so there is contact dripping of blood and fluids on surfaces. There is no level of separation between the customers and the market itself in terms of the produce. So, and also there are some illegal practice, including animals that shouldn't be there, including bats and other animals. So that seems to be the first level of contact between the virus and the reservoir, let's say a bat who came in contact with an host. But then it's not easy just to think that as insufficient, because in reality, in order for a zoonosis then to become an outbreak, then to become an epidemics and a pandemics, you need not only this jump, let's say jumping from an animal to a person, but you need a sustained level of transmission. So a sustained interaction, and then a sustained mingling of the population in order to start spreading from person to the other and so on. And therefore you can think that something like described by this picture would sustain something like that. Why not? I mean, it's possible. And possibly what also favored this is a very high uh, R. If you remember what R is the sort of transmission rate, and because it's a respiratory virus with high transmission rate, maybe it was sufficient at the beginning to give enough momentum to then sustain an infection that would spread all over the city, and then movement of people moving it all over the world. So here you've got three different cases of viruses. One that has got a very, very unlikely probability to get in contact with a human, but when it gets in contact with the human, has got anyway a very low transmission rate, but it's quite dangerous. Here you got an hemorrhagic virus that has got a again low transmission rate, but very high mortality rate. And here instead, coronavirus that is the opposite, has got a very high transmission rate, a case study of a very high transmission rate virus, but with a less mortality rate, luckily enough, because otherwise it would have been an even worse disaster than we've got now. So I would rather close here with a single invitation to you to think about what is called the One Health strategy. The One Health strategy, which you will be studying and working into this sort of environment, is a concept that is emerging more and more recently and is about integrating the human with our practice with the animal and environmental factors. So basically, thinking more and more about how we relate with the environment and our fellow animals. And in a way, not to destroy them for sure, and also not to cause damage, but to sort of live in a harmony that is enough to make sure that we minimize these outbreaks. And this goes together with quite careful surveillance and prevention of emerging zoonotic diseases. And this is something that the WHO is doing, the CDC, and many other organizations, even in England, and basically basing on the decreasing forestation, protecting habitat, improving surveillance, and improving hygiene and education. So thank you very much. And I don't know if you've got any questions that we can talk about.